Hello, my friends. Dr. Dan here. Hope you are doing well wherever you are at on this beautiful planet. Today on Science Made Simple with Dr. Dan, we're talking about the science of cellular absorption. And what I hope to do is to explain to you how here at 3 International we use absorption technologies and how powerful they can be and really how imperative they are in supporting human health. So let's get started. We first need to talk about the importance of absorption. So I've said it many times, but if you haven't heard it, really the core premise is you aren't what you eat, you are what your body actually absorbs. This is an important thing because the body has millions of chemical processes that it performs every single second. That's called the metabolism of the body. And it uses, because it's called the metabolism, it uses metabolites or molecules. And these molecules are not all the same. Some molecules are more absorbable than others. And so in order to be effective, they need to get into the body and into the cell. And the interesting thing when it comes to promoting health like we do, where we use science-based nutritional supplements, is not all the molecules that we use are readily absorbed. And it's a spectrum. Some molecules are readily absorbed into the cell. You really don't need to do a lot with them. And others are basically completely not absorbable into the cell. And others, they span that gamut. So you have some that are more, some that are less, and a lot that are in the middle. So if we want to really improve human health, we need to make sure that the molecules that we have in our formulations here at 3 International get into the cell. And that's not as trivial as you would think. And when you look at plants and the molecules in plants, right, these molecules are not readily absorbable because they're not water soluble. They're what we call hydrophobic. These molecules, they don't like water. And that's a problem as the body is between 60 to 80% water, depending on what part of the body we're talking about. The other thing we need to understand is the concept of bioavailability versus cellular absorption. And these are two different things. A lot of people treat them as the same and they use that word interchangeably, but they're not. And so let's talk about that. So when we talk about bioavailability from a science standpoint, what it means is how much of a molecule reaches the bloodstream after ingestion. So whenever you ingest something, whether it's food or a nutritional supplement, and food supplements, they contain molecules. That's the key feature, the key commonality between them. These molecules, then they have to be digested. So they're digested through the stomach. They hit that stomach acid, which is pH 2. So it's quite harsh conditions. Most of the time, molecules can get through that. What we do here at 3 International, when we talk about absorption, we have different absorption technologies that we use to help ensure that these molecules survive the harsh stomach acid conditions. But anyways, they get into the intestines and then they're absorbed into the bloodstream. And so this bioavailability is how much of a molecule reaches the bloodstream after ingestion. And that can vary. So we need to get it circulating in the bloodstream. That's bioavailability. And as the name implies, it means how much, when I ingest something, how much of it is available for the cell to absorb. But that doesn't actually mean the cell will absorb it. So basically what it means, bioavailability, is how much of a molecule gets into the bloodstream and it's delivered to the surface, the outer surface of the cell, so then it has the potential to be absorbed by the cell. And then the second part, cellular absorption, is pretty straightforward. Once a molecule gets to the surface of the cell, what percentage of that molecule can actually get through the cell membrane and get into the cell where it could then cause a biological response. Because a molecule, if it has health promoting properties, it's of no use if number one, it's not bioavailable, and number two, it can't get into the cell. So that's what we're talking about. For something to be powerful and be beneficial for human health, number one, it needs to be bioavailable. And number two, it needs to get through the cell membrane. And the cell membrane is tough. It's picky. It's particular about what it allows to get through the cell membrane or not. And that's good because the cell membrane is the basic structure of survival for the human body, right? Our human body is made up of molecules. Those molecules put together make up a cell, and then trillions of cells make up the human body. So this is what we need to do. Here at 3 International, our company is 3 years old. And one of the things that we wanted to do when we started this company, one of the pillars was absorption, because we noticed that there is this problem that these beautiful, powerful, health promoting molecules, they just aren't absorbed very well in the cell. And in my career, I've been studying cellular absorption for over 15 years. In fact, when I first started in science and technology, I was synthesizing pharmaceutical drugs. And at that point in time, I realized that the drugs that I was making, that they weren't bioavailable. They weren't being absorbed into the cell. So in addition to developing technologies to synthesize drugs, I also developed unique technologies to make sure that these drugs could get into the cell. And then what I've done over the years, as I transitioned into the proactive wellness industry, transitioned into using natural, beautiful molecules from plants, I noticed that absorption was also a problem. So I've been utilizing techniques to increase absorption of molecules for many years. And here at 3 International, what we recognized is that there's not a one size fits all. Molecules are different. They have different tendencies, different polarities, which means how strong the affinity of that molecule for water is or isn't. If a molecule loves water, it's called hydrophilic. If it despises water, it's hydrophobic. And again, there's a scale with these molecules. So what we realized is that we needed more than one cellular absorption technology. There are supplement companies out there and they use one technology and they utilize that for everything. Well, as good as that sounds, there's no panacea effect. There's no magic silver bullet, one thing that does everything. If we really want to make sure that the thousands of molecules in our products get into the cell, we need to use many different technologies. So here at 3 International, we use 12 different cellular absorption technologies, and you can see them here on this slide. And when we use these technologies, they work on a variety of different molecules with different characteristics, and the result is that these technologies help to support and promote both bioavailability and cellular absorption. So I want to talk about each of these technologies briefly to help you better understand about what this technology is, how it works, what products we use this technology in, and to just get a better sense overall of the importance of cellular absorption. The first technology is what we call our yeast glycoprotein matrix technology. Now we use this on the vitamins and minerals in our Vitalite product. And when we were formulating Vitalite, what we noticed is a lot of these vitamins and minerals, they are not bioavailable and they don't have high cellular absorption. And so when we were looking at different things of what could we do to encapsulate these vitamins and minerals, we found that there's this technology called a yeast glycoprotein matrix technology. And yeast glycoprotein there's molecules called glycoproteins. They're actually part of the cell membrane from yeast. And what we found is that when yeast make vitamins and minerals in its natural native state, what happens is these vitamins and minerals are surrounded by glycoproteins. And we saw that this was happening to a degree. There was a tendency for this to happen. And then what we found is that we can actually optimize that technology to make sure that these vitamins and minerals are surrounded by glycoproteins from yeast. And when you do this, you create a nice little bubble where now this glycoprotein matrix with vitamins and minerals on the inside can actually easily diffuse through the cell membrane. And that's based on one of the common dogmas of chemical biology. Is called like associates with like, meaning that if you have molecules that are the same, have the same characteristics, they just come right together. Molecules that have different characteristics, they repel each other. And so glycoproteins are part of the cellular membrane, not only in yeast, but also in humans. And so when you have a glycoprotein matrix and it comes up against the cell membrane, it's just basically it assimilates itself into that membrane. And then what it does once it assimilates itself is it pushes out those vitamins and minerals into the cell into the intracellular space, and lo and behold, now you've absorbed these vitamins and minerals into the cell, where they can travel to the different parts of the cell that they need to and increase absorption. So that's what we use here. And this is a great technology that provides enhanced absorption. It's natural, it's food-based, and it makes sure that these vitamins and minerals are properly utilized. And that's why when people take our Vitalite product, they feel a response because these vitamins and minerals are actually absorbed. The second technology is liposomes. And liposomes are commonly used in the nutritional supplement industry, and they're great. They're a great technology. Basically, the way that it works is you create a lipid bilayer, and you could see this figure here on the slide where you have two lipids, where you have the polar head and the tails, and they connect to each other to form this lipid bilayer. And inside that lipid bilayer, you can place bioactive molecules from plants. And the thing with liposomes is they're customizable based on the lipids that you use. You can make them different sizes. You can't make them huge, so they can't encapsulate proteins or really large peptides or anything like that. But smaller molecules from plants, you can encapsulate them and introduce them into the cell. And so liposomes are a common way of doing that. Molecules that are hydrophobic, that don't like water, they could sit right in that pocket inside that bubble, and it's a great way to deliver molecules. We use liposomes in our Purify, Revive, and Immune and our Eternal products. So you can see it's one of our go-to strategies. We like it. The science is well known. We optimize our liposomes. We use a different molecular composition, so it's not exactly the same as liposomes used in other products, but because of this, we have a highly efficient delivery mechanism. So we use liposomes as often as we can. And again, liposomes work for some molecules, but they don't work for others. And that's why you see we have liposomes in some of our products, but not in others. Now let's talk about micelles. Micelles are basically a simpler form of a liposome. So instead of having a lipid bilayer, we basically have a lipid monolayer, where we use a molecule that's called an amphipathic molecule. And basically what that means is it's a molecule that's long, and at one end of the molecule, it loves water. It's hydrophilic. And the other end of the molecule, it doesn't love water, it's hydrophobic. And so when you put this together under the right conditions, these molecules self-assemble to form a bubble, a structure called a micelle. And the great thing is, again, molecules that don't like water, they slide right into, they flee for the inside of this hydrophobic cavity, and they sit there, like associates with like. Inside the micelle is hydrophobic, so hydrophobic molecules will slide right in there. And it's a really nice way to do it. We use molecules called triterpene saponins, and we use molecules from Kiaha saponins, we use ginsenoscides, and we use triterpene saponins from Yerba Santa and a few other places to make these micelles. So micelles are a really nice way to do it. If you know how to do it, they form quite readily. And that's the key. You have to know how to do it. You have to use the right conditions. But we've been doing this for a long time. We know how to make the right conditions. You can't just throw molecules in there and expecting these structures to assemble. You have to have the right conditions. Okay, now we have adjuvants. And what adjuvants do is they make the cell membrane a little bit leaky, a little bit fluid, more fluid than normal, just for a split second. And when you do that, if you have molecules that are somewhat absorbable into the cell membrane, what you can do when you add adjuvants is they can quickly get into the cell. So when you destabilize the cell membrane just a little bit, these molecules can get right in. And classic adjuvants are piperine and menthol, piperine from black pepper, menthol from peppermint. So we use those, and we use adjuvants in our Revive GLP-3 and our Visage skincare collection. Adjuvants are simple, they're easy to use. Again, you need to make sure that the formulation is right for adjuvants. Sometimes if you're not formulating the product properly, even though you have menthol or piperine in there, it doesn't cause the same effect, because there can actually be ingredients that can counteract these adjuvants. And so you have to be careful there. You have to know what you're doing, but this is a really powerful approach. And it creates a window of opportunity. I like that word, a window of opportunity, so the molecules can get right through. Next, we use chlorophyllin. which is a derivative of chlorophyll. Chlorophyll is the molecule from in plants that give it its green color. Chlorophyll is vital for photosynthesis and a lot of other processes. But chlorophyll, due to its structure, and the structure that we use of chlorophyll is called chlorophyllin, which is stable. And what chlorophyllin does, you look at that molecular structure, you see those nitrogens there. Inside those nitrogens, that creates a ring where molecules can go in there and they can actually complex. So they don't fit inside a bubble in this case. What they do is they bind to this molecule and they form this connection. And because of this connection, chlorophyllin can go in. Now, chlorophyllin is a beautiful molecule. It's called an intercalator. What it can do, if you look at the structure of chlorophyllin, it's flat, it's planar. It's like a piece of paper. So when you look at the cell membrane, basically it's just like sticking a really thin piece of paper through the cell membrane, right? If you have a big thing, it's not able to get in, but a thin piece of paper could slide right in. And so we bind molecules to chlorophyllin like that thin piece of paper, and it can slide right through the cell membrane or intercalate through the cell membrane and into the cell. And so this is a nice technology that we use, and we use it in our Purify product. And the reason we did that is when we were formulating this product, we found that there were several molecules that the other technologies, liposomes, micelles, just weren't working for silymarin and some of these other molecules that we needed to get into the cell. And we found that chlorophyllin was a good way to do that. And so that's why our Purify product is green. The capsule is green because it's made of chlorophyllin. All right, next we have fulvic acid. This is also used in our Purify product. And the reason why we chose fulvic acid is an organic molecule. It's found in soil. So in soil, good soil, you have fulvic and humic acids. And these are actually the complexes that provide organic material for the plants to survive. So when we talk about organic soil, that's soil that's rich in humic and fulvic acids. And again, what fulvic acid does, it's kind of like chlorophyllin, where it forms this binding interaction. But this binding interaction is called a chelation interaction when it comes to fulvic acid. So there's a chelating process where they're bound together through a pretty strong interaction. And then what fulvic acid can do is it gets into the cell membrane. It can chelate and get through the cell membrane. And then once it does, when the pH of the cell outside the cell is different from inside, the conditions change, then it actually releases the molecule. And that's an important point when we develop cellular absorption technologies. It's one thing to have it bound and to get to the cell membrane and even to get through the cell membrane, but it's another thing to have it let go. You don't want it to hold on all the time, because even if it gets into the cell, if it's still holding on to that molecule, then again, you have an increased absorption. And so there's technologies that we use, triggers, switches, that when the pH changes inside the cell from outside the cell and conditions inside the cell are different, our cellular absorption technologies, they recognize that difference and they actually let go of the bioactive molecule so that they can then promote healthy absorption. Okay, next we have polyphenol complexation. We use this a lot. We use this, as you can see here in the slide, in our Vitalite, our collagen, Eternal Kinetic, and GLP-3. And adjuvants are used in GLP-3 as well. But this polyphenol complexation, what we do here at 3, again, we are molecular experts. And polyphenols are nice because there's hundreds of different types of polyphenols. Some people might say polyphenol, that's the British pronunciation. So whether you say polyphenol or polyphenol, I'm American, so I say polyphenol. But what we can do with polyphenols, because there's so many different types of polyphenols, early on in my career, again, when I was synthesizing pharmaceutical drugs and trying to figure out how to make these drugs absorbable, what we found is that there were certain polyphenols that we could use that would actually coat and surround the organic compounds, surround the drug in my earlier case. And when I started formulating nutritional supplements, we found that there were polyphenols that could coat other natural molecules that were not absorbable. And they do this through what we call van der Waals forces, or non-covalent bonding interactions. And the great thing about this is these form interactions that are weak to begin with, but when you coat them again and again and again, it actually forms a pretty stable complex that can get through the cell membrane. But the beautiful thing is once it gets into the cell membrane and the conditions of the cell change, it rapidly disintegrates. These polyphenols fall off of the bioactive molecule, and then the bioactive molecule can travel to where it needs to in the cell and exert its biological activity. So this is a natural approach. This is nature's smart strategy. And with all of these, what we wanted to use is we wanted to use natural approaches. Because here at 3 International, as you know, we don't use anything synthetic, and that would defeat the purpose, right? If we're using natural molecules and we're using synthetic technologies, that doesn't make sense. And that really doesn't even work. If we're using natural molecules, we need to find nature's natural approach. And so that's what we've done. It takes a lot of time sometimes. It takes a lot of effort to figure out how to do that, but that's what we do. Another approach is called hydrolysis. So sometimes molecules are just so big that they can't get into the cell. And collagen is one of those. So with collagen, we can't really use any of the other technologies we've talked about because it's a big molecule. So what we need to do with that big molecule is we first need to break it down into smaller and smaller pieces. And that's where our scientific absorption technology called hydrolysis comes in. And as the name implies, what we use is water and other natural ingredients. And what we could do is we could take this large protein and we could chop it up into smaller and smaller pieces. And these smaller and smaller pieces are called peptides. So proteins are connected together by a bond called an amide bond. Amino acids, as you know, a protein is hundreds and even thousands of amino acids connected together. And where amino acids are connected together is called an amide bond. And so what we can do is we can use this hydrolysis technology to break apart these amide bonds. And the cool thing is we can actually use other natural ingredients with water to make sure that we break apart at different amide bonds, specific amide bonds, so that we give and we obtain specific peptide fragments. And so that's how this works. We take a large protein, a large molecule, we break it up and we create peptides. And now these peptides, with their smaller size, can actually passively diffuse through the cell membrane. And again, we use this hydrolysis approach in our collagen product. A companion approach with hydrolysis, and we also use this in our collagen product, is called micronization. Because one of the things that can happen is that with this hydrolysis technology, we break the big protein into smaller peptides, but these peptides are lonely. They still don't necessarily like water. And so what happens is they'll glom onto each other. Even though they're smaller fragments, they'll glom on and complex with each other. And this is called aggregation. And so what we do is we do a further process called micronization. And what we do is we use energy and physical force and shear to disrupt the aggregation. We're not degrading the molecules or anything like that. We're basically using technology to separate them from each other. And when we do that, these small molecules, these small fragments remain apart and they can enter into the body. Because sometimes even with hydrolysis, if they're aggregating, it's still kind of like you have a big molecule and they can't get through the cell. So that's what we do, hydrolysis and micronization with our collagen product. Then we have what we call enzyme delivery. And how this works is we use different enzymes in our products. In our Vitality and Purify products specifically, we use enzymes. And we use some of these enzymes are actually used that the body uses to break apart molecules into smaller and smaller pieces. The body needs those for overall general health, but we also add enzymes as part of an absorption strategy. And the way it works is there's enzymes. Enzymes are designed, a lot of them are designed to break molecules into smaller and smaller pieces. And these enzymes are customized in the sense that one enzyme specializes in breaking apart a certain type of bond, like a carboxylic acid bond. Another enzyme specializes in breaking an amide bond. Another one specializes in breaking up a carbon-oxygen, a hydroxyl bond. And so what we do is we throw in specific types of enzymes to break apart small molecules that we can't use micelles on, we can't use liposomes on, we can't use yeast glycoprotein matrix technology on, but enzymes we can. And so this is another approach where we make smaller and smaller pieces, smaller and smaller fragments that then are able to get through the cell. And the hydrolyzation, micronization technology also works on them. And so we use complementary approaches, and that's how we do this. So enzyme delivery is used in vitalite and purify. Okay, then we also use another approach is chelated amino acids. So when you're using minerals like calcium and magnesium and manganese, a lot of these minerals are not absorbable in the body really at all. And a common form that people use in supplements like magnesium oxide and calcium hydroxide and whatnot, these are not bioavailable, number one, and they're not absorbable through the cell membrane. And so a lot of times when you see these ingredients used in a supplement, you basically know you're getting zero absorption. And there were some scientists about 20 years ago that figured out a really cool technology called a chelated amino acid technology. One of the reasons why these minerals are not absorbed is because the things that they're bound with are not absorbed either. And so these scientists found that if you took an amino acid and you took zinc and magnesium and put them together, what you had is you formed a magnesium bisglycinate ingredient. And this ingredient actually has really good cellular absorption. And they're covalently bound to each other. And what it's able to do is it's able to go through the cell membrane through what we call an active transport approach. So if we want to get through the cell membrane, there's two different ways. There's passive diffusion and there's what we call active transport. And these minerals that are in this chelated amino acid form, they're picked up by this active transport process and they get into the cell quite readily. So in our immune product, we use this technology to make sure that the minerals get into the cell, and it's a quite effective approach. And then the last approach, number 12, is what we call molecular shuttles. And we use this approach specifically for our topical skincare product, our Visage skincare collection. So absorption into the body through digestion and into the bloodstream is quite a bit different approach than absorption on the skin. And so we need to use different technologies to make sure that the ingredients in our Visage skincare line actually can be absorbed deep into the skin. And as you notice here on this slide, you have different parts of the skin. You have the stratum corneum, which is a surface layer. You have the epidermis, the dermis, and the subcutaneous layer. And if you want to make a powerful skincare product, you want to make sure that these ingredients absorb to the dermis. That's really where now these ingredients are able to transform and support healthy skin. And so the approach that we use is called a molecular shuttle approach. And basically what we do is we use ingredients like lecithins and caprylic acids. And when you use a certain combination and certain types of these, what they're able to do is they're able to form chaperone-like complexes around the bioactives in our skincare formulas, and they're able to shuttle them through the skin barriers so that we're able to deliver these deep into the skin. And like I said, this approach is unique and specific to our Visage skincare collection. Okay, well, this is all fine and good, but does it actually work? Well, here at 3 International, what we do is not only do we use new technologies, we demonstrate that those technologies actually work. And so we ran on our nutritional products, what we call our Simple 6. We ran not only a bioavailability, but a cellular absorption study. And we use what we call a CACO2 assay. CACO2 assay is the gold standard clinical study to show bioavailability and cellular absorption, more specifically cellular absorption. So what you can do is you can actually make a cell membrane through a really cool technology. And then what we do is we place our ingredients on top of that cell membrane, and then using analytical instruments and a technique called spectroscopy, we can actually detect what percentage of the molecules on the outside of the cell membrane are able to get through to the inside. And that's how we calculate percent absorption. And what you can see here on this graph, on the far right of the graph, we use a control. We use a curcumin supplement because there's a lot of curcumin supplements, and curcumin has a similar molecular structure to other molecules that people use in nutritional supplements, and they're not highly bioavailable. And what we found is that on this control, only about 10% of the molecules that we put on the outside of the cell surface got into the inside. And then we compared that with our Simple 6, our foundational health metabolic products. And what we found, you can see the results here, is that our products were anywhere from 3 to 9.5 times more bioavailable and more cellular absorbable than the control. So this is evidence that these technologies, these 12 technologies that we're using, not only do they sound good, not only do they, in theory, make sense, but in practicum, they're working and they're increasing the cellular absorption of the beautiful molecules that we have in our products. So with that, I hope that I've shown you that what we do here at 3 International that's different from any other company is we use a multitude of different cellular absorption technologies to make sure that all the beautiful molecules in our products can get into the cell where they can support healthy gene expression, healthy epigenetic function, so that they could then cause or elicit a beneficial response in the body. Thank you, my friends. Sure appreciate you. This is Dr. Dan signing off.